Good morning students. In the previous class we discussed about paper electrophoresis and gel electrophoresis. Now the new topic is STS PACE. So we already learned about PACE, polyacrylamide gel electrophoresis. So when a PACE is attached to the STS, we called it as sodium dodecyl sulfate polyacrylamide gel. So it is widely used in different chemical laboratories like biotechnology and also biochemistry and also in the molecular biology laboratories. So it is used to separation of different proteins. So here when anionic detergent STS added to the paste, the combined procedure is termed as the STS paste. So here we will use the anionic detergent. So here, due to the masking of the charges of proteins by the large negative charged on STS binding with them, the proteins migrated along the gel in order the size. So basing upon the size, the molecules will be move in this culture. Okay, so here STS is an anionic detergent which denatures the secondary and non-disulfide bridges and makes the proteins or amino acid into rod shaped structure, and it is easy to move freely into the gel. So it converts the net negative charge to the polypeptide in the portion of its length. A polypeptide chain binds to the amount of STS base, and the portions is relative to the molecular mass. So observe this picture. It is the STS base. In this we have one buffer tank. So in this buffer tank we will use the buffer. So we having this here spacers and having the negative and positive electrodes, and we have the glass plates. In the spacer we can prepare the gel beads. Okay, so first one is stacking gel, other one is separating gel. So first we have to add the separating gel. So first, by using polyacrylamide gel we can prepare the separating gel. So here big proteins and small proteins are separated one by one. Then we can add, first we have to add the separating gel or running gel, then we have to add the stacking gel. So before adding the stacking gel we have to pour some of the water, and we have to add the next gel, that is called as stacking gel. So we can use dyes in both of the gels for different in two different types of gels. So then we have to add the comb. So here when we are going to add the comb, we can observe some of the wells. So then these wells are loaded with the samples. Then these electrodes are going to attach it to the current power and going to generate the electrophoresis. So now we can observe different types of bands. So here larger fragments are there and the smaller fragments are also present here. Okay, so observe this picture. This is the process of preparing the STS base. So then next we can get different types of bands in it. So during this process we can get different types of bands, and the process of electrophoresis process is going to be held. So by using glycine buffer, it is present in the upper reservoir. It is present in two forms, as a Zwitter ion and the glycine anion. After the gel sample is loaded, the power is switched on, and the chloride proteins and glycine anions starts moving to the anode. So after entering into the stacking gel, the glycinate ions have low pH, and so they are converted into Zwitter ion state. So the Zwitter ions will not move any side. They will be in the mobilized state. Immobilized state means they will not move. They will be stay at one place. So the immobilization of glycinate Zwitter ions is the higher mobility chloride ions and create high voltage. Okay, so during this preparation or getting this high voltage, the proteins have mobility which is the intermediate between trailing and leading ions. So the proteins move quickly towards the chloride ion and forms a bands. Okay, so here we will observe different types of bands by using different types of dyes, bromophenol blue. The bottom of the gel currently is switched. When the colored gel is come bottom of the gel, we can stop the current. The gel is removed. Then the gel is removed. The gel is taken in a stain solution like commercial brilliant blue for few hours and washed. The proteins are visible as blue bands on the clear black background. So this is the process of SDS space. So first we have to add some of the colors. Okay, so we can add bromophenol blue. So when the bromophenol blue come bottom of the reservoir, we can stop it, and we have to separate the proteins which are going to be given as the sample or identification sample. That is the process of SDS space. The next one is applications. It is used to estimate molecular weight of proteins and nucleic acids, and also determination of subunits of the proteins, and also used for the isolation of proteins, and also monitoring the protein content in the body fluids. And in the body we can contain many number of proteins. That proteins have to be monitored by using this technique. During this we have different types of proteins like globular proteins. So these globular proteins also can be observed or monitored by these electrophoresis, and it is also used for the bonds which are present in between proteins, that are disulfide bonds, can be estimated or identified by using these electrophores. And it is also used for the quantifying. The number of proteins can be observed, and blotting applications also. Blotting means the preservation of some of the proteins, nucleic acids like RNA and DNA. So for these purposes also we can use these electrophoresis. Next topic is isoelectric focusing. It was first discovered by Svensson in Sweden. So it is based upon the resolution. It is having high resolution, and it will separate the plasma proteins at least 7 bands. So here the proteins are separated in a pH gradient according to their isoelectric point. So we will say basing upon the isoelectric point only it will be run. So basis principle involved in the electrophoresis is isoelectric point. So here proteins are subjected to the electric field in the pH gradient, and it also requires the solid surface, as normally we can use the polyacrylamide. So first we have to know what is meant by isoelectric point. We also said it as a PI. That means the pH at which the net charge on the protein becomes 0. So below PI, this means positive charge. Above PI means negative charge. So here proteins moves towards the electrode with the opposite charge. So if having the positive charge, if the proteins having the positive charge, they will move towards the negative charge. If the proteins have negative charge, they will move towards the positive charge. So during this motion proteins will either pick or lose the protons. During different from conventional electrophoresis migrate to steady state. So this is the picturization of how the molecules will be moved from one place to another place basing upon their electrophoretic mobility. So here they will be charged or they will be moved from one place to another basing upon their pH. So the molecules which are having the middle or stationary, or that we will say stable pH gradients, they will be stand up in the middle portion of the system. The remaining charged particles, if they have the positive charge, they will be moved towards negative charge. If they having the negative charge, they will move towards the positive charge. This is the main composition or main principle based or behind upon this chromatography. So here all the proteins have an isoelectric point of pH. So this will be if we take a mixture of proteins that can be electrophilized through a solution that having a state pH gradient into a form anode, to that the cathode, and the each protein will migrate to the positions in the pH gradient according to their isoelectric point. So this is called as isoelectric focusing. So here this is the picturization of the isoelectric focusing. So if we take a negative charge, so that having the pH 9, they will be moves towards the positive charge having the pH 3. So an ampholytes are used in this solution which is incorporated into the gel. So first we have to know what is meant by ampholytes. So these are pH gradients that will establish the pH to the molecules which are going to separate. Here we will use 600 to 900 Daltons, which are closely spaced ionic charge and high conductivity. So the curve is determined by pH and interval covered by the ampholytes and the distance between the electrolytes. So first how the procedure will be taken. So first we can use the acrylamide solution, water, ampholyte solution and ampholyte urea, and spin gently to mix urea and add 10% of APS and the timid at the end, and we have to remove some of the bubbles and fill the cascade completely with the solution and allow to polymerize at the room temperature. So first we have to take the chamber, then we can add the some of the solutions along with the water, and we have to adjust the pH of 3.5 to 10, and we have to use or add the urea for it. Okay, so after that urea preparation, we can spin the urea or mix the urea with the above ampholytes. So then we have to add the 10% of APS and the timid at the end of the procedure, and when we are mixing, so many air bubbles will be obtained, so that air bubbles will be separated from the that mixture. So then fill the cascade completely with the solution, then we have to close it and polymerize at the room temperature. Then we have to add some of the distilled water and centrifuge the sample to remove the aggregates, so which the clumps are formed. To remove that clumps, we can use the centrifuge, and then apply the supernatant to the wells within the disposable tip of the Helmland syringe, or we can use micropipette for the applying the supernatant to the wells. So run the electrophoresis at the 150 volts for 30 minutes, or we can use 200 volts for 2 hours. So this is the procedure to follow the isoelectric pH. So here we can use the ampholytes and pH gradients for the separation of different molecules based upon their pH. Okay, so this is the process of isoelectric pH. So first, during that we can observe different types of bands basing upon their pH. We can observe different sizes of the bands. So here how we will prepare the gel during the separation of electrophoresis. So when we are adding the gel, we can use the combs for making the wells, and we have to add the sample for the separation. So this is the process I already said. So we have to add the samples with the equal volume of 2x loading buffers, and loading buffer includes the loading buffer containing some of the components like urea and ampholytes, so which is having different types of pH, and we have to add the mercaptoethanol, and also to assess the difference between bands we can add some of the bromophenol blue dye. Okay, then this is the process, and we can separate the bands by using fluorescent and UV absorbance. We can detect the samples which are present in the electrophoresis. Okay, that is the process and the detection. The next one is advantages. What are the advantages for the proteins? Are proteins, when the proteins are there in the composition, we can obtain the proteins in different forms and the little as 0.00 pH units can be separated. For the protein separation we can use that much of pH. So as spreading of the bands is minimized due to the application of applied field, and the pH gradients high resolution can be achieved. So isoelectric focusing is a powerful method for the separation of specifically for the separation of the proteins, and we can use to separate the serum proteins also into 6 different bands. So performing IEF is easier because the placement of the sample application is not important. Then what are the disadvantages in the isoelectric focusing? So here limited stability of the solutions is the present, and also inadequate purity. The substances which are going to separate, they will not be in the purified form. That is one of the disadvantages. So here we can use high concentration of ampholytes, and high voltage will be necessary. So that is one of the disadvantages of using isoelectric focusing. So then applications. So it is used for, I already said, it is used for the separation of proteins and also peptides, and we can use the type of proteins like serum proteins, and we can use globular proteins and also fibrous proteins. So and it is used in the research of taxonomy and cytology. Highly sensitive electrophoretic technique. So here it is particularly useful to study the microheterogeneity in the proteins, and it is a method for particular useful for the separation of enzymes which are different forms of same enzymes. So because of its convenience, speed, polyacrylamide gel plates and isoelectric focusing is most commonly used in clinical and forensic sciences and also human genetic laboratories of serum proteins. So this method is used in the food and agricultural industries. So in the agricultural industries we are used it for the separation of different molecules. So this method is also used for the research in enzymology. So during we have different types of enzymes. So during the process of enzymology we can separate different types of enzymes which are using in these equations. So that enzymes also separated. So because of that it is also used in the enzymology, and also it is used in the membrane biochemistry. So here membrane biochemistry means we having different types of materials in the membranes. That membrane containing enzymes or some of the proteins that also can be observed or separated in this technique, and it is also used in the microbiology and also used in the immunology. In the immunology we have different types of complexes like MHC complex, cytokines. These MHC complexes and also cytokines also observed in this technique, and it is also used in the cytology and the taxonomy. So here cytology means the cells. Okay, the study of cells is called as cytology. So we having different types of cells. During that cells we have to separate some of the cells for the experiments. So these cells also separated by using this technique. So we call it as it is also used in the cytology and also in the taxonomy also. So taxonomy means the separations of different types of plants based upon their families. So these also observed in this electrophoresis. The next one is pulsar field gel electrophoresis. So standard gel electrophoresis, it is a standard gel electrophoresis. It was unable to separate very large molecules of DNA. So in this technique we will use two different types of electrical fields. So one is normally generated and the other one is generated spontaneously when the DNA fragment is moving from one side to another side. So here we will use or we will separate the DNA molecule at the large size of 15 to 20 kb. So here the migration will be occur essentially move together based upon the size. So at Columbia University in 1984, David C. Schwartz and Charles Cantor developed a variations on the standard protocol by introducing an alternative voltage. So alternative voltage means we will use normal voltage, then suddenly we will use the another voltage of high voltage on volts having. This technique is become known as pulsar field gel electrophoresis by using these two different types of or alternative forms of voltages. So here pulsar field gel electrophoresis is a technique which uses the separation of large DNA molecules by applying gel matrix and electrical field that periodically charges or changes the directions. So if we have two fragments which having highest molecular size and the other one have lower molecular size. So first when we apply the voltage, the two molecules are going to separate. Then when we are applying the high voltage, the smaller molecules will take low time for the movement and the larger molecules will take highest time for the movement. So during this the separation will be occur. So here the technique is used by the scientists to generate a DNA fingerprint for a bacterial isolation. So here pulsar fields means what? Any electrophoretic process that use more than one electrical field alternatively. That is called as pulsar field. So here these molecular scissors are selected to generate a small number of DNA pieces that can be separated based on size. So usually these DNA pieces or restriction fragments are large and needs to be specially treated and separated to generate a DNA fingerprint. So first the bacteria are loaded into the agarose suspension similar to a gelatin. Then the bacterial cells are open to release the DNA. Once the DNA is released, then the agarose and the DNA are going to separate. Then these are treated plugs are then loaded onto the agarose gel and the restricted fragments are separated based on the size using electrical field. So what makes the PFGE different from how scientists usually separated DNA in is because PFGE can separate several large fragments and small fragments by using the restriction endonucleases to this electrical field that constantly changes directions to the gel is based upon generating is used to generate the DNA fingerprinting. So here this is the process of pulsar field gel electrophoresis. So first we have to take the bacterial culture. In that bacterial culture we can by using agarose gel electrophoresis we can separate the fragments. So and then we have to place it in the DNA is now in a plugs. Then we have these plugs are inserted into the pulsar field gel electrophoresis. So here the scientists loaded the DNA gelatin plug into a gel and spaces in the electrical field and separates the DNA fragments according to their size. We can use data analysts to know the separation of the different bands. So the gel is stained so that the DNA can be under ultraviolet rays or we can use UV absorbance spectrophotometry. A digital camera takes a photograph of the gel which having different types of bands. We can observe it in the computer.
Then applications. So here southern and northern blottings. So the complementary of two strands of DNA provides us with an unique method to find whether the DNA can be mutated DNA or the normal DNA which we are interested is present in the sample or not. And the sample DNA is separated by agarose gel electrophoresis and transferred into the nitrocellulose paper. And then the complementary radio labeled nitrogen base sequences of our interest is allowed to react. So by using gel electrophoresis, that means pulsar field gel electrophoresis, we can use to do the southern blotting technique and also northern blotting technique for the storage of the DNA.
and it is also used for the determination of relative molecular weight of some of the DNA fragments. So as the separation is based on their relative molecular mass of unknown DNA fragments, and it is identified by comparing with the molecular mass of the standard DNA fragments. So this is one of the application for the pulsed field electrophoresis. And the next application is maybe it is also used for the genotype and genetic fingerprinting. So here genotyping means we have to know the genetical character of the substances. Basing upon these analysis, we can give the genotype of a organisms, and we can give and we can going to use in the genetic fingerprinting. So when there is some genetical issues, there may be in the family, or by some of the thieves which are going to take in some of the robberies, and some of them in murder cases, we can use this genetic fingerprinting. Who are the suspected, we can observe. So in that process we can use this isoelectric pulsed field electrophoresis. So and it is also in this we can commonly consider a gold stands. So in epidemiological studies of pathogenic organisms, so the pathogenic organisms means that are microorganisms like bacteria and viruses. These viruses and bacterial epidemiological characters can be separated or observed by using this pulsed field electrophoresis, and also used for the subtyping has made easier to discriminate among the strains of Listeria monocytogenes. So here we can observe some of the clinical manifestations during the separation in the foods, and also environmental contaminations in the food can also be observed, which are done by Listeria monocytogenes. So these are some of the, or these are the major applications in the PFGE. Okay, the next one is advantages of PFG, that means advantages of pulsed field electrophoresis. So it is a subtyping has been successfully applied to the subtyping of many pathogenic bacteria, and has a high coordinates with the epidemiological retardedness. So PFG is the basic format, can be applied as the universal generic method for subtyping of different types of bacteria. That means subtyping means the major group of bacteria, and we are also having the subtypes of bacteria. That subtyping of bacteria can be typed or assessed by using PFGA. So here we can use different two types of alternative voltages. During this process we can separate or assess different types of microorganisms also. So here this electrophoresis method is to optimize it for the each species. So these are we can also separate different types of species. These are most of the important advantages of PFG, and we can observe DNA restriction patterns during the generation by using the PFG are suitable or stable, and also reproducible again. Okay, this is the advantages of pulsed field electrophoresis. The next one is limitations of pulsed field electrophoresis. So they do not really know if the band of the same size and the same piece of DNA are present in the medium or not. That will be really we cannot observe. That is one of the limitation. And bands are not independent, they are dependently they are formed, they are not independently. So the retardedness should be used as a guide, not true phylogenetic measures are present. We have to use some of the guides or relatedness or related organisms. Basing upon the related maps or relating ratios we can observe. Some strains cannot be typed by PFG. Okay, so that means in the, if we take a group of bacteria, in that we can observe only some strains of bacteria. We will not completely study all the strains of bacteria in the PFGE, and it also require trained skillful technician. So if the person who are using that PFGE, they should be skilled person. So if there is no skill in that person, it will be not completely purified form. So and does not discriminate between all unrelated isolates and can't optimize separation in every part of the gel at the same time. It will take more time from one gel and at the distance of some places, and it does not discriminate between on all unrelated species or isolates, and it is the time consuming pattern. Thank you.